Okay, so I'm one of those tall people that we have to accommodate. So I just wanted to say thanks for the opportunity to kind of talk to you guys today about research in general, and thanks to Matthew for the fantastic introduction, and you'll get quite a different look at research. And Matthew spoke a lot about research, but a lot of it was clinical research. And so what we're looking at is what's happening inside motor neurons at the basic molecular level, and how that affects that clinical phenotype and that misfiring or hyperexcitability that Matthew was talking about. And this is just the beautiful coastline of Wollongong. And so we have a research team in Wollongong, MND Research team, and we're trying to recruit as many researchers as possible, and different, I guess, different looks, and we're trying to learn from other fields as well. So we think the more people working on motor neuron disease, the faster we get to something that might look like a cure. And my clicker is not working. I'm not sure why. I'll just point. And this is just to say thank you to a lot of people that contribute to the work that we do and contribute to motor neuron disease research in general. And I just wanted to say a big thank you to Professor Bob Brown from the University of Massachusetts, who kindly loaned me some of the slides that I'll show you today. He's one of the world experts in motor neuron disease research. So what I wanted to do, I kind of want to give you a picture of what it is to do motor neuron disease research and what I mean by motor neuron disease research. Talk a little bit about how it all fits in and how all of this research can potentially work together towards getting a therapeutic into the clinic. And Matthew has already done a fantastic job about talking to you about motor neuron disease and the amyotrophic lateral sclerosis. And as he pointed out, we've known about this for quite a long time. And I'm going to switch pointers. And I'm not having any luck with pointers. No. That's working. It's just me. It's not the pointers. It's just me. Okay. And this is the same quote, and Matthew and I did not coordinate this, but this quote here was made by Charcot in the 1860s, and I think that this same message applies today. And as I said, this is in the 1860s, and we're still searching for something that is going to really make a huge impact on the lives of people with motor neuron disease. And as Matthew pointed out, there are the two aspects, those upper motor neurons and those lower motor neurons from the spinal cord, is a big focus of research. And another aspect that Matthew pointed out was this focal onset. So some part in the motor cortex where we get a seemingly random site of onset, and then this kind of spread throughout the motor cortex in the brain and up and down the spinal cord. And we think this is a really important aspect of the disease, because what that tells us is this is like a domino effect or a bushfire effect, and so it starts in one place and then kind of spreads out from that location. And that's important to us because that gives us something to target, because if we can stop this spreading of the disease, then this gives us some kind of tractable target to aim for. And this concept has been a lot of years in the making, and a lot of people didn't believe that this was actually occurring over a lot of years. It took us a long time to convince people. Another important aspect, and although Matthew said that there is a dying forward hypothesis, there's also a dying back. And so one of the first molecular changes we see is a removal of that connection between the muscle and the spinal motor neurons, and we see a dying back of those really long axons. And the other molecular changes that we do see are these, what I will call accumulations of junk protein. And this is a really old image, maybe from the 1980s, and these dark spots here are what I would call junk proteins accumulating and clogging up the motor neurons, which is a focus of our research. And this also occurs in the in these long axons, we get these accumulations and these big bubbles. Okay, so the normal axon will be this nice little thin stripe here. We get these big bubbles of accumulation, basically blocking these spinal motor neurons. And so, at least in our laboratory, we are focused on following these accumulations. And so I'm just going to show you what this looks like. So you can see this is actually a motor neuron that we're studying in culture. And you can see these little blobs starting to accumulate in this nerve cell. And we can follow these through time. And eventually what you will see is, and this is followed over in the lab for over 72 hours. And you can see that at the end, this motor neuron just basically explodes because of these accumulations. And when we look into spinal cord, you can see these are, I'm showing almost like a heat map. And so where we see cell loss in the spinal cord, we also see TDP pathology or these accumulated proteins. So we think that they're a really important part of the process. And most of our study is on the inherited forms, and this is because we know some of these genetic mutations that cause motor neuron disease, and that gives us something to research and something that we know, whereas the sporadic disease is much, much harder to study. We have known about familial forms of MND since the 1890s, and it's through these kind of genetic families that we can get a lot of information. And so, as Matthew pointed out, the first mutations that were known to cause MND were in this SOD1 gene, and that was back in the 1990s. And the next genetic discovery didn't come until some way into the 2000s. And then you can see since then, with the increase in technology, we've increased the number of genes to over 35 different genes that we know mutations are associated with MND. And this is a list of all of these different genes. So we consider these genetic abnormalities, we consider them pieces of the puzzle. So we can, if we can understand what each of these genes does, then we can start putting that puzzle together to build a picture of what's happening inside motor neurons. But not all of those genetic changes actually cause MND. So there are a small number of genes in this list here that we know mutations in these genes cause MND. And especially something like SOD1, the mutations in this gene give a really strong predisposition. It's what we would call highly penetrant. So if you have a mutation in SOD1, then you are very, very, very likely to get motor neuron disease. And similar with all of these genes here. But these other genes in this much longer list, if you have a mutation or a change in those genes, then what that means is that you have a predisposition. It doesn't mean that you will get MND, but you have a predisposition to motor neuron disease. And these genetic changes, the more that we know about modern genetics, the more changes we will find. So there are already really large studies being undertaken, and this particular one, which was published earlier this year, what they did was that they basically sequenced the entire genome of almost 3,000 people with motor neuron disease, and they came up with a list of changes that they thought represented what motor neuron disease was. And we really do need to increase this, as Matthew said, to 15,000 to really extract the information out of this. And so they found, as well as finding changes in genes that we already knew about, they're also finding changes in new genes. And that's important for a couple of reasons. It's important for us as researchers so that we can research these changes, and it's also important for the clinic because they can give people a definite diagnosis if those mutations are within the family. And so we are still, there's still a big question mark as to what the cause is of most of sporadic motor neuron disease, and a large proportion, which is getting smaller and smaller every year of the inherited forms. But we do know quite a lot about the genetics, and so greater than 60%. But 20 years ago, this was much, much lower. And as Matthew pointed out, there really is an overlap between a range of diseases now with motor neuron disease. And in this big oval shape here, all of these genes are associated with motor neuron disease. But then we have all of these other overlapping little circles here, which represent other disorders such as frontotemporal dementia, Paget's disease, some of the lower and upper motor neuron disorders, and mitochondrial pathology. And you'll see that there are overlapping genes across all of these disorders. It really is, the more we know, the more complicated this genetic mix-up is becoming. Okay, so what does that information tell us as researchers? And I don't necessarily expect you to know what these details mean here, but for us in the lab, we're starting to cluster these genetic changes into groups, into functional groups that we can kind of identify. And essentially, what we can then do is find that these genetic lesions are causing certain changes in certain aspects of the metabolism of the cell. And these are the things that we're chasing down. So you can see that there is a really large grouping here, and those changes we think are representing dysfunction in the movement from the genetic materials, which is the DNA, to a signaling molecule called messenger RNA, which is a copy of a gene. So there's a really large number of genes that fall into this category. And there is other, such as transport of molecules along those really long axons is another, I guess, category that we're kind of combining here. But we think that changes in these functional or metabolic pathways all kind of contribute to motor neuron disease. And we're trying to understand how these genetic changes feed into changes in these functional pathways. And as I said, so in the clinic, motor neuron disease looks very similar and has a pattern of changes, and we're really trying to understand how the changes in these genes result in these changes that we already know about. And so one in particular, and this is all cartoons of molecules that are bouncing around inside the nerves, but essentially all of these red circles here represent genes that are associated with MND, and you can see that they all kind of fit into this schematic diagram. But essentially what this means is that one particular, we think, way that a motor neuron can die, motor neuron disease, is through a faulty mechanism for clearance or degradation. It's like, I guess, taking out the trash. So we have natural garbage trucks inside each of our neurons that basically will degrade faulty proteins or aged proteins. And when this is faulty, we get an accumulation of these junk proteins, and this is the image that I showed you at the start. So now there are a number of genetic mutations that suggest that not only do we see this pathology in motor neuron disease, but there may be a genetic element to that, and that this is actually a really important mechanism. And so there are now trials undergoing to see if we can kind of speed up this removal process, that maybe this will kind of rescue this accumulation and rescue motor neurons. But it's never really that simple, and this is just a diagram from a paper that someone has put together, all of the proposed ways that these genetic mutations might harm a motor neuron, and you can see that there are many things going on, and so we really do have to kind of understand the underlying defects that cause all of these complicated changes. So one thing that I wanted to kind of point out, and Matthew did say that this particular mouse that they use, the SOD1 mouse, is not a great model. But one thing that we have been able to do in this model is to identify this disease progression. As I said to you, there seems to be this spreading of pathology, but there was never a really good model for this. Some researchers in the States took some really sick mice. They took the spinal cord and took a bit of that spinal cord and injected it into in the spinal cord of another mouse that although had some of these human proteins that cause MND, didn't get MND. And what they found that when they injected even a tiny amount of this material, that this mouse went on and got MND. And this is not to say that this is an infectious disease. What this means is that it needs a trigger. So, and this was the injection of this material from another mouse. And so basically, if we know that it can be triggered and then that there is this progression or a domino-like effect, then we should be able to stop that. But we're still trying to understand exactly how that process is communicated between neurons. Another huge part of MND research across the world at the moment is on this genetic mutation called C9orf72 that Matthew mentioned. And there are a lot of different theories about how this is killing motor neurons, and it could be a gain of function, something that it wasn't really supposed to do before, but the mutation changes that. Or it could mean that it's losing a function. It changes the protein in such a way that we no longer have it working the way that it should. And I'm not really going to talk about all of these intricate little diagrams here, but just to say that this is a really hot topic of research, and we think that this is actually, because it's now the largest known cause of MND, so that there is a lot of research in this particular field at the moment. And so another way that we study, to study MND in the lab, we need to have a model of disease. So we need to be able to get down to the cells and have a look at what's happening inside cells. So obviously we can't do that any other way but have models, and that's either in a dish with cells or in an animal such as a mouse. And another way of studying motor neuron disease is in the dish. This is something that we are doing in Wollongong, so with Dr. Lizanne Uy. And essentially what we can do here is that we can take a little skin sample from someone that has MND, and we can trick those skin cells in the dish. We can grow and they'll multiply in the dish, and we can trick them into becoming stem cells. And so we call these IPSCs, and so that means induced pluripotent stem cells. Pluripotent just means that they can then be converted into any kind of cell in your body. So we take these skin cells, we trick them into becoming stem cells, and then we feed them a whole range of different growth factors, and they can convert into being motor neurons. And we can sort of study these motor neurons in the dish, in the lab. And so this is becoming a really important tool, because for us, we believe this is the only way to study what's happening in the neurons of sporadic MND patients. So before this technology, we had to have a genetic model, but now we're able to study what's happening in sporadic MND with these models, and we're just starting to do that. So I can't really comment on what we're finding just yet, because it's very, very early days. And they are very lengthy experiments. I told you before that we do some experiments which take over 72 hours. These experiments take a couple of months just to get these motor neurons, and then we can start to look what's happening inside those motor neurons. And another model organism that we're starting to use, and this is at Macquarie University, is zebrafish. And see, these little fish are just ordinary zebrafish that you can buy at the aquarium, except that we can implement genetic changes in the brains of these fish that can mimic motor neuron disease. And that's important because the real reason that that's very, very important is actually we can actually peer in. These fish are totally transparent, so we can peer into the brains of these fish and see what's happening in real time. And so this is an image. These green, I guess, neurons that you can see are motor neurons inside the fish. And here is a neuron that we're going to watch. And this happens over about 10 hours, but essentially what you'll see here is that that motor neuron dies, and we can watch that. And so these mice are these fish. They're not furry fish. They are just normal fish. And so we can actually watch an individual neuron dying. And so we really think that we can understand what's happening in the brain, because otherwise we're just looking in a dish. And here we can peer in directly into the brain and spinal cord of these fish, and we can't do that with mice either. So we think this, again, is going to be a really important tool to kind of understanding that progression of motor neuron disease. So how does all of this research, and what I've described to you has been different types of research, I guess, and different things going on around the world, and how does that, what the important question is, how does that get us towards a therapy? And I think that this is, for me, this is the way that I look at these types of different aspects of research. So we really need to look at the genetics and understand what's happening at that level. We identify genes that cause or give us a predisposition towards getting MND. And then we need to take that information and then get into the lab and look at either a model, like an animal model, like a mouse or a fish. We do a lot of cell culture, so we're looking at individual neurons and what's happening inside of those. We can take the skin biopsies and grow patient neurons for sporadic MND. And then we need to feed that into and make sense of all of that information and understand how all of these different changes affect a neuron, so that we can then come up with something that we think can kind of alter this complicated changes in the neuron. And so this is how we hope that our work is contributing to getting closer to a treatment for MND. And just to give a couple of examples of where some of these treatments are at. And so this was work published in 2006, but the clinical trials are ongoing. And this study was performed in the mouse, and this is a genetic therapy, I guess you could call it. And so essentially it's what you might call an anti-gene, and so this is in the SOD1 model, and essentially it will lower the amount of all of the gene, not just the mutant. So it is a complicated story. You have to be careful not to lower it too much, but we know that when you lower the amount of this gene, especially the mutant form, then you get a better survival. And this is in mice, and this curve this way, the black one here, tells us that those mice are surviving longer when we lower the amount of this gene in those cells. And the trick here is being able to deliver it. So they have done a phase 1 clinical trial with these anti-gene type therapies. And so hopefully in the next 12 months, there will be a phase 2 trial in this type of a therapy. I think this is a really promising therapeutic, specifically for the genetic forms, because in those cases, we know what the genetic changes are. We're yet to understand whether or not the same genes will have such a beneficial effect on sporadic patients. And this is just depending on when. This is a different form of this anti-gene, and you can see a much longer delay so it gives an extra 69 days, and over that life of that animal, that's an additional 50% lifespan for that animal. So this is a really encouraging potential therapeutic. Again, we don't know how this will translate to other forms of MND apart from the SOD1. But the C9orf, for example, is another target for this particular type of therapy. So it really is a promising avenue. I also wanted to speak about some other research, and this is from Melbourne, and a drug called copper ATSM. Again, it's in the MND mice, but essentially it's the theory behind this particular drug. The researchers in Melbourne believe that there is a copper imbalance within the neurons, and they are delivering very, very carefully with this drug a tiny amount of copper to motor neurons and seeing a beneficial effect. And so again, this particular drug and the research is meaning that hopefully by the end of the year they'll have a clinical trial on this particular drug. And just to point out that there are, across the globe, there are around about 70, 75 clinical trials, based on the preclinical work that we do in the lab, around about 75 clinical trials going on. And this particular website here shows the list of clinical trials that are going on worldwide. And what I've done here is just kind of summarize those trials so that you can kind of see the way that this is panning out. And so I've kind of categorized those clinical trials into different areas that relate to the type of research that we do. So there is a number of trials with looking at growth factors and how that protects motor neurons. There's only a very limited number of gene knockdown trials at the moment, but nerve stimulation, there's a few trials. Stem cell therapies, there is, at the moment, there's quite a number of stem cell therapy trials. And immune modulators is another area which is quite heavily being trialled. And if you look at where in the world these trials are happening, you can see by far the most trials are happening in the US, France and Germany, and then there's a few trials scattered around the rest of the world. There is no, on this particular database, there is no Australian clinical trials. But so I wanted to end with this story so that all of that research that we're kind of feeding into the world literature is based on kind of getting some therapeutic targets and getting drugs into clinical trials. And so that's how I'm going to finish